Good afternoon, everyone. My name is Dr. Erika Ganda, and I am very happy to be able to showcase the breadth and depth of work that's going on here at Penn State. And it's my great pleasure to introduce today's speakers and people whose research is going to be highlighted. So as you will see, we have Dr. M'ikanatha. He's from the PA Department of Health. He's going to be talking about the human health side of things. I'm going to touch on the work that Elizabeth Ransom did. She unfortunately cannot join us. Then we're going to have Dr. Veronica Roman Reina. She's going to share some of her AMR work. Dr. Elizabeth Hines unfortunately could not join, so she sent a video that if times allows, I'm going to play at the end. Otherwise, I can just share that and we can put it on Exchange. Dr. Nicole Ginan is going to come up right after me, and then also Dr. Edward Dudley is going to talk about his food safety and molecular microbiology. I just wanted to highlight all the different things that we have here at Penn State that relate and pertain to antimicrobial resistance. So one of the things that really draws our attention here is the transdisciplinarity of the work that we have at Penn State. And this is possible because of the buy-in from all the departments, the colleges, and the Penn State Hook Institutes of the Life Sciences through the One Health Microbiome Center, which you'll learn a little bit more. So the way we look at this thing is we work on a One Health perspective, at least from my lab. But before you can have human health, you need to be able to have healthy animals and a healthy environment to produce crops. And the way I look at this is the glue that actually keep all these three health domains together are the bacteria. So we need microbes for digestion, we need microbes to clean up environmental spills, and so forth. So that brings me to my part of the research at the Ganda Lab. We focus more on the agricultural health sector of One Health. And the way I like to depict my lab and research program is something like this. We're really excited about antibiotics and microbes, and we're species agnostic. I did start on the dairy value chain, and I studied mastitis, which is the inflammation of the mammary gland. If my which is the inflammation of the mammary gland. And that's where I got bit by the antimicrobial resistance bug and got me really interested into microbiomes and things like that. I looked at in different ways, both in regular infection, so like experimentally infected and also naturally infected animals. Then I moved on to a postdoc where we looked at the milk microbiome from a food safety perspective in partnership with IBM. And that started with having to isolate DNA from milk, which is really challenging. So that turned into its own paper. And the AMR part of things in machine learning is currently about to be resubmitted. So my work here at Penn State started in July 2019. And again, the laboratory's goal is to develop microbiome-based solutions for use in the agricultural industry. One of the big drivers of our research program is antimicrobial resistance, and that is mainly because there is a lot of consumer push for raised default antibiotics or no antibiotics ever, and there is a very big gap in knowledge when it pertains to what are the alternatives that we can use, how can we keep our animals safe while using the least amount of antibiotics possible. So this is the Ganda laboratory, and below I have the picture of the Penn State Microbiome Center in 2019, which is a fantastic collection of folks that Nicole's gonna cover. And this is how we looked like in 2023. So we've grown a lot. We have a really big breadth of expertise all around microbiomes through the Tree of Life. My team is comprised of 5 current members and 4 alumni, and you will see by the little icons below, we are species agnostic. We've had protocols in 7 different species. I'm trying to reel that back in. Of course, I'll never stop doing dairy research, but I'm really interested in food production systems and on One Health perspective and how can we do things to make everybody else healthier. So I just have this slide to highlight some of the work that my students are currently working on. Sophia Kinney is focused on Salmonella doubling and genomic epidemiology, looking at antimicrobial resistance patterns, looking at the role of dogs in antimicrobial resistance transmission with humans. Dr. Janine Lechon is a reproductive specialist, and she looked at the antimicrobial resistance and microbiomes on the most common diseases on the 3 species that she works with. So there will be metritis in the cow, endometritis in the horse. And we also are starting to do some antimicrobial resistance profiling of the female dog microbiome. Dr. Ana Fonseca is a veterinarian and she's interested in applied research. So she's the one going to the barn and feeding 3,000 chickens different things so we can understand what do they do to the antimicrobial resistance profiles and profitability. And last but not least, Samantha Sabel is our most recent graduate student. She's a second year, and she's interested in genomic epidemiology, global health. We have some work that is ongoing with Kenya right now, and her main project at the moment is on improving a method that we developed for targeted antimicrobial resistance profiling. I want to give a shout out to Dr. Elizabeth Ransom, who reached out, and I have had the luck to collaborate with her for a while now. And basically, we can do everything we want to the animals, but in the end of the story, it's usually the humans that are the ones choosing to treat or not treat. So we collaborate with Elizabeth Ransom in social science to be able to better understand what are these drivers and human and ethical dimensions of antimicrobial resistance in dairy farms. Now, kicking back to us, I'm going to be happy to hear from all the other speakers today. Nicole, you're up.
Great, thank you. I guess I'll just share my screen. Hopefully it works okay. How does that look for you? Are you seeing presenter mode? Oh, it looks good. Okay. So, hi, I'm Nicole Ganan, and I'm currently the research project manager for the One Health Microbiome Center at Penn State. So my background is in agricultural microbiome, sustainable ag, and thinking about how we can develop alternatives to using antimicrobials in the field, such as microbial biological controls. And I've worked in everything from high-valued perennial tree crops like citrus, all the way to just cash crops, corn. So I have a pretty big breadth in thinking about like the different approaches and different risks within those separate systems. But today I really want to focus more on my current focus and talk about AMR from a microbiome sciences perspective, and also give a highlight of the center. Let's see. Okay. So our center is the first microbiome center to formally take on a One Health integration. And we live at this intersect between One Health approach and microbiome centers. And really, the special part about this is that we have a very broad expertise that covers human health, environmental health, and agricultural health. And from our perspective, the microbes are the base of the biosphere, and they flow across these systems to connect the health of the globe, of everything. Our center is directed by Seth Bordenstein, and we also have a 14-member executive committee that represents our broad expertise. So we have people from plant sciences to art and architecture, a faculty member from the medical school, Erika Ganda, who you just heard from, and even anthropology and so on. So this diverse group really helps to drive and direct the center, to be inclusive and continue to think about our One Health aspirations. A little bit more about the center. We are currently serving around 540 members, and that includes everybody from undergraduates, graduate students, postdocs, staff, and faculty. More specifically, we have 125 faculty that represent 42 different departments, again, highlighting our very broad expertise within the center. And we have award-winning and just fabulous faculty and trainees. And importantly, we've started to move a lot more into not just research, but the educational realm of microbiome sciences. And we have the very first microbiome sciences dual title PhD. Here at Penn State, it's the only place in the world where you can have microbiome sciences on your PhD title. So we're very excited about that. And we have this huge community, over 500 people, and we provide a lot of different activities and resources. So I'll just highlight a few, community building, different resources. We work to develop research working groups. So we have all these different researchers from different departments who maybe are working on similar questions in different systems, and they're not, sorry, we'll answer the questions after, yeah. Who maybe these people wouldn't have talked, but since they're a part of the center, we can bring them together and work on solving these larger problems. Different educational workshops, fellowships, and we prioritize outreach as well. So getting back to more what the microbiome is and the purpose of the center more broadly is we like to think about microbes as the biodiversity within the biodiversity. Again, seeing them as the base of the biosphere and supporting health and life of all systems. And a lot of times when people talk about antimicrobial resistance, they're really focusing on bacterial pathogens. But I want you to think more broadly about microbiomes in general, which include bacteria, fungi, viruses, and other microorganisms. And when we say microbiome, it means all the microbes that are living and interacting in a specific environment. All these microbes interact with a host potentially, so a human, an animal, a plant, or with each other. And they can range from being helpful to harmful, but often are neutral. And of course, all these different interactions are context dependent. So you can have one bacteria where maybe in a baby it's harmful, but in an adult it is neutral. And other other things that can impact the lifestyle or the interactions of microbes could be temperature, environmental conditions, as well as nutrients. And we do see this a lot. So again, just adding to the complexity and thinking about not just something that's a recognized pathogen, but risk that could be associated with something that could turn into a harmful microbe under certain conditions. And just to further kind of explain the complexity that I like to think about when I'm thinking from a microbiome perspective. So here I have a simple three strain microbiome. The first microbe here produces a secondary metabolite that is inhibitory to this orange microbe here, and that keeps it in check, in balance, and keeps the population down. This orange microbe, produces a byproduct that this blue microbe can use as a carbon source. So it actually promotes the growth of this other microbe. And this is, a balanced microbiome, miniature microbiome. A pathogen comes in and we notice that and humans want to tackle it. So we target it with an antimicrobial. And of course, we have that risk of the AMR developing. And this is, where I see a lot of AMR research is mostly focused on this. But let's say, no, the antimicrobial works perfectly. The pathogen is gone, but we end up having some off-target exposure and antimicrobial resistance develops in the orange microbe. And potentially this inhibitory secondary compound from the strain that keeps this strain in check now no longer works. Maybe it has a similar mode of action as the antimicrobial that we had applied to take out the pathogen. So now you start to see that the green is being outcompeted, and you begin to see ecological consequences far beyond what was expected. So you have an increased population of this specific orange strain, and then that increases the population of the blue strain, and you start to have all these huge changes in this natural microbial community dynamic. Oops, let's go back. And so this is kind of when I'm thinking about AMR, I'm thinking about the broader dynamics and how that changes. So now this microbiome is much different than its original natural state. And how does that change its function? Is this microbiome now a pathobiome and can make people sick? Is it not functioning in the way that it needs to? And these are the larger questions that I think we need to think about more. Then bringing it back, I also feel like we have 125 microbiome faculty, and I would say that very few of them feel comfortable talking in an AMR space, particularly our plant researchers, even though they are microbial experts. And a lot of this is because I found that microbiome research field and the AMR research field are fairly separate, which doesn't necessarily make sense. So I kind of want to advocate for doing these things together. And of course, the two major things that are roadblocks in both of these fields is education and awareness, and then doing more research and understanding and doing risk assessments. So what I would propose is, for instance, in a clinical setting, we can team up. When we start to talk about, good microbes exist, and you have gut microbes, and there's probiotics, and different food feed your good microbes, you can take that into account when making a decision on either taking an antibiotic or not, because you're not just saying like, okay, if you take this antibiotic, you're potentially risking way down the line AMR, right? And then people don't necessarily respond to that because it's so hypothetical. But if you're saying, well, you're having this potential risk to have antimicrobial resistance development, but you're also could be and will likely be negatively impacting your healthy, good microbes. So now you can have that kind of double story to really make people think about whether this is an appropriate time to use an antibiotic or not. And then on the other side of that, a lot of research that is happening in the microbiome world, we use a lot of metagenomics, which is whole genomes of whole communities from all different places. And these microbiome researchers are not looking at antimicrobial resistant genes, but they already have that data because they have the whole genomes. So I think that more collaboration and repurposing of metagenomic data will be really important for driving both of our fields forward together. And then I just wanted to, advocate for developing these collaborations with the amazing trainees and faculty that are working in our microbiome center right now. So here's just a picture of some of the variety of different systems that our graduate students are working in, from oceans and terrestrial areas, from crops and livestock to mice models and even ancient DNA. And I would say almost all of these grad students have AMR gene data within their metagenomics data, but they're not really looking at it. So if you are interested in kind of like taking this collaboration forward, feel free to reach out to me and I could connect you with some different researchers who might be working in systems that you're interested in. And then lastly, I also want to advocate for and mention our One Health Microbiome Symposium that's coming up in May 30th through 31st. And again, this would be an excellent place for microbiome researchers and AMR researchers to come together and think about think about a One Health approach, but also collaborations and how we can support each other. Yeah. So that's all I have. Hopefully I didn't go over too much.
No, thank you, Nicole. That was wonderful. Ed, do you want to share your slides or do you want me to?
Yeah, I can share. Thanks. I think, yep, there we go. All right. Well, thank you for this opportunity. Let's move a couple of things out of the way here. All right, so what I'm going to share with you today is just a kind of a short story about a nascent project in the lab that's kind of developed with over the last couple of years with some collaborators in Puerto Rico. And, you know, as Erika was talking about, her big background is the dairy chain. We're actually, you know, moving into that a little bit. But before getting into that, I just want to introduce the current group. Going across the in the back row in the left, we have Zoe Goldblum and Aaron Horak, who are the two undergrads who are in the lab, who are doing a lot of work on trying to look at the presence of foodborne pathogens that can be monitored in wastewater treatment facilities. Zoe actually has a pretty cool paper that just got posted to bioRxiv, showing that the Salmonella Septemberg that was responsible for the 2022 peanut butter outbreak could be isolated from wastewater treatment plants. Moving one over, Sharon Nieves Miranda, who is been working on projects around identifying new O antigen groups for E. coli. Expect her to graduate this semester. Anna in the top right, we'll come back to her in just a little bit. Bottom row, Erin Naraki, who recently left us for the University of Pittsburgh. She did a lot of work on E. coli 015787 colonization in cattle. Middle row is Jay Feng, is a master's student who's graduating this semester working on E. coli 015787 genomics. And Edwin Lopez, on the bottom right, who's doing an interesting collaboration with the University of California, Berkeley, asking if all that we have is a genome sequence, can you make a prediction about what bacteriophage that strain is going to be sensitive to? But the big project's going to be the one for Anna's, who's coming back in the top row. But before we get into that, just kind of giving you a quick theme of the lab, we're interested in asking questions that basically require your very first step to be to sequence a large number of bacterial genomes with a particular focus on E. coli and Salmonella. And I just highlight, some of these papers and recent trainees, also indicating, Erika had mentioned how collaborative of an environment, Penn State is. You notice another speaker who's going to follow, Nkuchia M'ikanatha from the Pennsylvania Department of Health, has been an instrumental collaborator on all of these projects for, probably going on about 8 years now, Nkuchia. So the project I'm going to tell you about actually began as part of a different program that we run that I direct, and it includes 4 other faculty members here at Penn State. That's an REU program, stands for Research Experiences with Undergrads, and basically means every summer we bring in about 8 to 12 undergraduates from different colleges and universities around Pennsylvania, from the University of Puerto Rico, basically students who are at institutions where they really don't have access to cutting edge research tools. We bring them in for the summer. We teach them about hypothesis generation and about doing research. And at the end of the summer, they get to present their results. And in 2019, another long-term collaborator in the upper right of this slide, Dr. Jadira Malaves Acevedo, who's at the University of Puerto Rico, she sent these 2 students below, Sharon, who I've already introduced. She's the one who's finishing a PhD in my lab, and Paula Lopez, who is at the Ohio State University pursuing a PhD, sent them up for the summer program with a bunch of E. coli. That Jadira has a program in her lab where she has undergraduates go out to farms, isolate E. coli from different animals, And we thought it would be interesting to build this into a summer project doing some genome sequencing. So Sharon and Paula arrived with these strains, particularly bringing strains that came from both from calves and from cattle. And the calves were fed on those farms using two different treatment methods. Either they were getting milk from bulk milk tanks, or they were being fed waste milk, which is basically milk that comes from animals that are being treated with antibiotics that can't be sold on the retail market, but they can be used for feeding calves on these farms. So they brought these isolates here. We did some characterization. We found out about 52 of them truly were E. coli. Took a random selection of 32. So 8 of each of these categories. We did whole genome sequencing and phenotypic AMR. So it was a really nice, concise, 8-week project to learn about genome sequencing, learn about AMR, and how we can correlate these two. So the results that they got were pretty interesting, although, admittedly, when you go back into the literature, we actually, should have expected this, in which what you're looking at are the 4 different category of E. coli isolates on the bottom, and each of the boxes in this bar chart are different antimicrobial resistances that were identified in various strains phenotypically. And basically the bottom line is from the farms that were feeding the tank milk, the bulk tank milk to their animals, all E. coli from these cattle were pan susceptible. A little bit of resistance in the calves. From the waste milk farm, we had more resistance in the adult animals and a dramatic increase in the calves. I said, this has actually been shown before. We know that E. coli tend to be more antimicrobial resistant when they come from calves versus adult animals. And also there have been other studies that have shown, feeding these animals waste milk does have this impact. So the students, sequenced 32 of these E. coli isolates, and basically this is a table to show, again, as we've kind of known with E. coli, with salmonella, that from a genome sequence alone, we can make a pretty accurate prediction about antimicrobial resistance from the genes that we identify in the genome. So just kind of demonstrating again that we can, for most of these antibiotic classes, that we were able to predict resistance from the genome sequences. So, Sharon and Paula went home, and Jadira and I got talking a little bit more about, well, is this something we want to try, we could make something of, or is this a one and done type of a study? And we started looking a little bit in the literature, and one of the things that was really kind of striking to us is there's been very little surveillance data about antimicrobial resistance at all in Puerto Rico. And in fact, 2 years ago, we only found about 8 papers that were associated with food, soils, or water. And what was eye-opening to me is that dairy is actually the largest economic driver of agriculture in Puerto Rico. I would have thought it was something a little bit more tropical. $172 million in 2017. There's almost 300 dairy farms on Puerto Rico, and the majority of them are situated in the northwest part of the island. We connected with Dr. Jaime Curbelo, who's in the bottom right, who has an active extension outreach program in Puerto Rico around mastitis, particularly caused by E. coli and Staphylococcus aureus. And he basically told us how this is a really big issue on Puerto Rico, because even though they are a U.S. territory, they're required to follow U.S. laws. Just like a lot of other places in the world, there's still a really big problem with the availability of antibiotics to farmers and the fact that they're using these directly to treat their animals, kind of in a non-evidence-based, non-scientific manner. And he was telling us that many of his clients will run into a situation where after a couple of years of just self-medicating their animals, they pretty much have to cull their entire herd because they've had so much antibiotic resistance going on in E. coli and Staph aureus that they can't treat their animals who have mastitis anymore. He's also got some pretty cool studies, showing he's got a positive correlation between how much training that he provides to these farmers and the milk quality that can be quantitatively measured in a variety of different metrics. So the 3 of us kind of came together, and after a second try, we were able to get some funding through the USDA in their antimicrobial resistance program. So this is something that just started up this past August, which we're kind of taking three prongs. The one on the left, kind of continuing some of our surveillance. We're going to be looking at 20 farms twice a year in their hot and cool season, or as I've learned what the Puerto Ricans refer to it as, their hot and damn hot season. Dividing farms up by high quality milk and low quality milk as well, which again can be quantitatively measured. Doing some characterization of some of the E. coli Staph aureus isolates that we find associated with mastitis around the farm, and also collecting a lot of information about some of the practices on these farms as well. All the way on the right, we're building a cohort of 16 undergraduates who are going to be actively involved in this program, helping to not only get the isolates, do some of the whole genome sequencing, they're going to learn how to do bioinformatic analyses, participate in some national and regional forums, and really the idea being we're not going to really tackle this program in Puerto Rico unless we can kind of educate the next generation of leaders in this field. And lastly, in the middle, Jaime is going to help develop some train the trainer programs, but really, I think what we're going to focus in on is not so much the training coming from the university, but can we identify leaders who manage farms that have high quality milks and teach them to how to train some of their peers who are having some some problems on their farms. The idea being they're probably going to be a lot more effective at creating lasting attitude and practice changes. So with that, just again, got another picture of the lab. Everything the research that I talked about, again, that's funded by the USDA. Other programs in the lab are funded by the FDA and the NSF. And I guess I will turn it back over to you, Erika.
Thank you, Ed. Veronica, can you share a little bit of your work with us?
Sure can. Let me share my...
This is like dating across Penn State, pretty much.
let me see if I can share my screen. Stop to share. Do you see the presentation mode? Or awesome. Okay. So first, I would like to thank Erika for inviting me. As I say here, I'm barely new at Penn State. I started in July last year as an assistant professor. And I consider myself a plant microbiologist. So that's my training. So I mainly focus on plant pathogens. But lately, since there is so many in-house sequencing tools, I was trying to shift a little bit of my research of how we can make these tools approachable and also use them for disease diagnostics. So how we can use genomics or even metagenomics to use that as tools for diagnostics. And that's kind of what I want to highlight here, of some of that ideas that I have in my lab or I'm building on, is on how we can do microbial dynamics in agricultural systems, along with surveillance. And what I would like to talk more about is part of that genomic epidemiology of plant pathogens. And with that, what I would like to idea is that I'm going to explain a little bit of kind of why I get into antibiotic resistance, and one of the questions that I think are answered, and kind of I think this is a great opportunity to integrate different disciplines to try to answer and have a more overview idea. So to give you a background, because I know not all of you work with plant pathogens, is that we consider two types of treatments that we use for crops. Are either people use heavy metals such as copper to treat some diseases that happens in vegetables, but we also use antibiotics such as streptomycin and tetracycline to treat diseases on trees. And these two usually have some bacteria that are well known to be plant pathogens, but also we have an idea of what are the genetic elements associated with that. So I think we already understand what kind of are the genes associated with that, but kind of what would be the next step, what we do with that information. And that's kind of why I get into this research. Is I'm going to show you a little bit of what I have been doing last year, and it's associated with a disease that has been an outbreak in geranium. So here we have what a pretty geranium looks like and what an ugly geranium looks like. So usually for ornamentals, you don't want to see this kind of leaves. And that is actually caused by a bacteria called Xanthomonas hortorum, which, as you see here, it has like a chromosome and a plasmid. And I was trying to use genomics to understand why we see an outbreak, why now. So since we have, as I mentioned, in-house sequencing tools, I was able to analyze the chromosome and the plasmid separate. And what was striking is that we see that in the chromosome, we don't see much changes since the 70s to now. So we see the same chromosomal structure. So we start looking now into the plasmids. And that's kind of, and goes to my, I got more interested because it's where we can use this kind of in-tool or in-house sequencing tools such as MinION. And with that, I start looking into the plasmids of strains from different years. And we see, for example, that from the 70s to 2022, what happened is that we see a reduction of the plasmid. But what I would like to highlight your attention is on the yellow region that is associated with metal resistant, in this case, copper resistant. So that's what I start looking at more into how these genes have been moving around these crop systems, and I start looking what could be the origin of these genes. And one of the we already known that some of these genes is already present in different Xanthomonas pathogens in the plasmids, but we start also seeing that some bacteria that are not associated with plant pathogens, and is mainly on the chromosome, also have these genes, which aligns with, I think some of you were already mentioning, that we should probably start looking at the context of how we see these genes. And one of them is probably some of these genes are already in these crop systems, and we are just selecting so the bacteria can acquire them with horizontal gene transfer. And one of them is actually from Stenotrophomonas, that we see that some of the genes are actually similar in the plasmid. So because of that, then I start looking in what happened to all Xanthomonas. And this is some of that idea on the right side, where we see that some of other Xanthomonas hortorum in the yellow area are also having these copper-resistant genes. And what is very interesting is that we see that it's across different years or decades, even different continents and different crop systems. So we see that we start asking questions more about what is the agricultural system that is allowing this movement to happen, or how, mainly what are the mechanisms that we're actually helping to have this happen with the systems that we're using. And see, for example, some of them are associated with greenhouse conditions. And this is only one part, where we also see that on trees as well. And even some other bacteria that I didn't put in this figure. So that's kind of, now that I have seen this part of my research, something I'm gonna start focusing now is, can we predict these evolutionary trends of resistance? Since some of them are acquired by horizontal gene transfer, how we can start tracking that as well? Can we identify the sources? So we know that the soil, for example, can be a huge reservoir of these genes. So how we start analyzing that and keep that in mind when we're trying to do treatments with antibiotics in different crop systems. And also something that I would like, I'm gonna try to work as well, is how we can use artificial evolution experiments to see if we do several passes of that bacteria, how is the rate of the movement of that genes? What are the key elements that we need to keep in mind when we're doing these antibiotic treatments? And probably most of you are also thinking of the same questions. So that kind of brings me to my last slide, and it's kind of how we can integrate that. So I see this is a great opportunity for that. My expertise might be more on the crop side, but probably all of you have the same ideas in different systems, or probably are the same mechanisms as well, or even the same genes, right? So how we can integrate not only in the agricultural system approach, but then how we can start integrating that ideas with livestock and food science. Of course, behind all of this is humans, so I didn't explicitly put it in here, but kind of having that idea first, and then can we I don't know if it's too ambitious, but then can we do more as a national way where we can use these technologies such as, for example, genomics or metagenomics to start keep tracking that antibiotic resistance across the US first, and then expand to even other continents as well. And that's it. Thank you.
Thank you so much, Veronica. We're so excited to have plant people join the group. It's been a long time. We've been looking for that. Talk about human people research. Nkuchia, would you like to share your screen and share your research with us? Maybe Nkuchia is having some technical issues. While he comes back,
Oh, sorry, Erika. I'm not having any technical issues. I just want to bring the slides.
go ahead.
Okay. 1 second. I just need to go to the folder with the slides. Okay. 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 Just. Can you see the slides now?
No, it looks a vertical.
What about now? Okay, let me see. Stop sharing and then I will start sharing. Okay, what about now? Okay, let's see.
I have your slides if you want me to share them.
Okay, one second and then I'll tell you if you should share them.
And then while Nkuchia works on that, Veronica, there is a open question for you on the Q&A. You can just type your answer there if you'd like.
And then we'll also address the questions vocally at the end of the presentation as well, so that those who are not on this webinar can view it in the recording.
Sounds good.
So are you able to see the slides now? Erika, do you see the slides?
No, you're not sharing screen, Nkuchia.
Oh, you're saying I'm not, okay.
I'm just gonna share mine.
Okay, that's fine, okay.
There you go. Just tell me when to go next. Everybody can see it?
Um,
Can everybody see the slides now?
Yep, looks good.
All right, Nkuchia, go ahead.
Okay, yeah, let me, okay. All right, so I have an appointment at the College of Ag and also with the state. So I'm going to present to you from the human side. If you go to the next slide. So I don't have for disclosures. The work that I'm presenting is funded by the state, FDA, and also CDC, and also support from Penn State. But I am presenting all these views as a scientist, and I am myself responsible for the views I express. The next slide. So when we think about antimicrobial resistant, it's important to look at them in terms of what are the burden of antimicrobial resistance in humans. So can you bring the first slide, Erika? Do you have another slide before?
No, that's it.
Yeah, okay. So if we look at antimicrobial resistant in the United States, for example, 2.8 million infections occur each year. And of those, we have over 16 cases per 100,000 are associated with salmonella. So we can use salmonella as just one example of the impact of antimicrobial resistance, and then I will share some of the study, at least just one example of the study that we are doing to better understand the impact of antimicrobial resistance. So nationally in the US, over 200,000 cases of AMR infections are reported through the public health systems. And of those, 6 of them result in death. Go to the next slide. So one of the questions that I ever ask, and there are no answers, is usually in the clinical guidelines, there are statements and also national agencies such as the CDC that salmonella usually causes mild infections. And sometimes they are severe based on the type of patient and some characteristics. However, the antimicrobial resistance aspect hasn't been fully described, and also it's not clear the sources of data that show the burden of salmonella and also what type of salmonella. So here we began a study, and Dudley is involved in this study, as well as one of the clinicians in Hashi and microbiologist in our lab, in the health department lab, people at CDC and the FDA. And what we are interested in is really understanding what impact that salmonella, if it's drug resistant, has, or even if it's not drug resistant, has on patients. So to conduct such a study requires a lot of agreements and effort. So we received a data use agreement. Then it had to be approved by the IRB at Penn State, because here we are going to be looking at the actual treatment records. Next slide. Next slide, Erika. We conducted this as a prospective study, and it also involves isolates that are submitted to the Department of Health. And these isolates are usually characterized. We know what cell types they are. And so we use that as part of the starting point. I'll go to the next slide. And based on the records that we have of the salmonella isolates that are reported, we brought that back to Penn State and said, these were the isolates that you reported. And now we have to collect treatment records for these patients. And that is where you have to have an IRB, and one person is authorized to look at the data and do abstraction. So we had to abstract records for treatment based on the agreements that were done. And these data were put into a database. Next slide. So the type of information that we selected to be able to characterize the patients were demographic characteristics. We looked at their past medical history. Then we looked at what the diagnosis was, and then the antibiotic treatment. And we also looked at the severity of infections, whether they were hospitalized, went to the emergency room, and how long they stayed in the hospital, whether they went to ICU. Next slide. Okay, so we looked at the antimicrobial resistant, and for the isolates, because we already have the isolates within the public health system, and these isolates were characterized at the CDC, and some of them were characterized at the FDA. And we also characterized some at the veterinary lab here in Pennsylvania. Go to the next slide, please. So this is just an example of what happens when people have salmonella. So of the 160 patients, actually 82 patients, that we were able to find their medical records. So this figure here shows the total 82, and then the 57 that received antibiotic treatment. So of the people that received antibiotic treatment, there were 57 of them, which was 70% of all 82 that were that we had both the isolate and their medical records. So what you can see is that antibiotic, whether they were changed after the proper diagnosis, and that is one of the issues, one of the concerns that prompted this study, is that we hypothesize that usually treatment is not aligned with the actual infections or the resistant patterns of the bacteria. So here you can see that although antibiotics were changed in 79, it means 79%, it means that 21% after it was known what the diagnosis was, they were not changed. And so for after susceptible results, meaning that they were not drug resistant, so 23% were changed, and the follow-up antibiotic change was 21%. So what this means is the need for antimicrobial stewardship, and these are actual patients, and this is actual treatment data. Go to the next slide, please. So I said that we do characterize this as salmonella. Salmonella are actually not all created equal, and some of them cause a wide range of severity of illnesses, and some of them are known to be highly drug resistant. For example, Salmonella infatis, Salmonella typhimurium, sometimes, depending on where it is isolated. Go to the next slide, please. And this actually also illustrates that all of the patients treated at the hospital associated with this university, Penn State specifically, the multi-drug resistant isolates, of those isolates that we had, was close to 10%. Those ones that were resistant to I can't say the name, but trimethoprim sulfamethoxazole is one of the drugs that is used, and it is very a useful drug, as well as ceftriaxone. So the one that's a DSC is actually an oral drug. This is ciprofloxacin. And that one has been increasing, but here we don't show any trend. But of these patients, about 16, close to, okay, we can say 15%, those ones who are resistant to this drug, which is for adults, is usually preferred because it is an oral drug, it's a cipro drug. Go to the next slide. So to conclude, the medical records, if that is done, they can provide very useful information as to the severity of infection for Salmonella, and also the treatment patterns and the potential risk that is involved. And also, as all the speakers have mentioned, this work is usually done through a collaborative effort. This was just a snapshot. We have other work that we have done, looking at it from the One Health side and actually taking the sequences from these patients and comparing them with the ones that we have isolated in animals. One of them that Dr. Ganda works on is Salmonella Dublin. So among these isolates, there was a Salmonella Dublin that was linked to one that was isolated in cows. So it is really a One Health approach, and something that is should continue to be approached in a multi-faceted approach. Thank you.
Fantastic. Thank you so much, Nkuchia. So I hope with this 50 minutes, we're able to showcase everything that's going on here at Penn State. Of course, nobody had the time or the opportunity to go in depth. And now, if you want to take on the lead and field questions and such. We are very appreciative of the opportunity to NIAMRI, and we are willing to collaborate.
Yeah, thank you guys for your wonderful presentation. It seems like you truly have a One Health focus at Penn State, and it was wonderful to hit on every single one of those sectors here in our webinar. And so we did have a few questions come through, and we'll give a few moments to let some others populate as well, but wanted to let everybody know that our April session for Member Spotlight will be featuring the International Society of Companion Animal Infectious Diseases. And so we're excited to learn more about that organization. And then also kind of taking off of what Veronica mentioned, and my screen is not progressing for some, there we go. We have the continuing education opportunities at the NIAMRI Annual Conference, which will be held in Columbus, Ohio, May 14th through 16th. We've listened to some very interesting research snippets today and discoveries, and so those would be great examples of the types of abstracts we're looking for our conference in May. So if any of you speakers would like to submit an abstract, we'd love to learn more about that at the conference, or if it inspired anyone in our audience to share their research and recent work as well. We'd enjoy seeing your applications for lightning talks and poster sessions. And then also wanted to let you all know that though Dr. Elizabeth Bronson was not able to join us today on this webinar, she will be a featured speaker at the conference. She'll be speaking on bridging the gaps in interprofessional practice and behavior change. So we're looking forward to that. All right, so now that we have gone through that, I'd like to start with some of our questions. So this first one is for Ed. Are there any other U.S. territories that might have AMR issues on their dairy farms? Are there lessons learned from Puerto Rico that can be used to inform training for dairy producers in general? So multifaceted question there.
Yeah. I mean, it's a great question. I do not know enough about, Guam and other territories about how big is dairy production there as well, to know if what we're doing here can translate there. As opposed to the lessons learned, is that we're still really early in this, that this project just started in August. So it's still way too early to say we've learned anything that we can use to translate to other dairy producers. But I think kind of as we get together as a group on this project, kind of the hope is we can make some arguments for some of the farmers that need to improve that. Hey, by doing so, it will help, improve the quality of your milk and make an economic impact on your farm. Because I think in the end, it's going to be some economic driver that's going to be needed to drive changes.
Yeah, great. Thank you. Our next question is, how do you manage data security and privacy issues with accessing medical records for AMR data? I think that one was for our last presenter, if you wouldn't mind sharing some insights.
Yeah, so that usually is very, very highly regulated by the IRB oversight. So only one individual is allowed to abstract the data, and they are simply collecting the data by records. Of course, they have to have the names, but once that's done, then they put the data in REDCap without any identifiers. So none of the investigators, except to one person who is a physician, has access. Being a physician does not allow them access to the data in that way. The IRB allows that access, and it's easier for someone already in the system to be able to abstract the data. But yes, so the data are abstracted without, in an anonymous fashion. There isn't any information that is kept, and that is very, very highly restricted, as is in every study that involves clinical data, and they have to be done. That's how we know vaccines are effective or drugs are effective. Those studies are allowed in the United States, but they have to be conducted with a full IRB review and monitored all the time. Thank you.
Yeah, thank you. All right, and then Veronica, I know this was addressed in the questions, but do you have ideas for how resistant genes might travel from, for example, France to North America? Would plant-based products be a source of organisms that can carry resistance genes?
So I can talk about specifically for some ornamental crops where what they do is they grow some of the plants in different countries and they import it to the US. So they do cuts, that is kind of already you have a small plant and then that's how you move them around. And I know some places have like big greenhouses and that's how they import the material to the US. And another big hot topic now is also the seeds as a source to move part of that microbes around. For plant-based products, I don't know what they mean because yeah, you can have DNA there, which might have sources for that, but it's still just because you have DNA, that doesn't mean it's gonna transfer to a live bacteria, right? So in that, I will be more cautious on how to talk about that area, but I'm no more aware of the plant cuttings.
Great, thank you. And Nicole, what is the most surprising thing about One Health microbiome research that you found?
Yeah, so similar to my answer, I think it's very surprising how when you talk about One Health or microbial One Health research, it's all about pathogen spillover and of course like COVID and etc. But what we should really be thinking about too is that there's this whole variety of microbes, including beneficial microbes, that we don't understand how they impact across systems or what functions they might have, or, every day we might be encountering something that is positive for our lives and our function, but we don't even know about it. And I just mentioned like one example that there was a lot of headlines about it, how microbes could be the next antidepressant and whatnot. But there's a soil bacterium that they found that actually when you breathe in its volatiles, it can increase serotonin levels. Yeah, so it makes you think about why people who garden might be happy. I don't know. And think about too that, with pets, we're exchanging microbes with them all the time. And there's studies that show that your microbiome becomes more similar to your pet as you have them for longer. And there's all these different exchanges that could have positive or beneficial effects. So thinking about a One Health approach very broadly is kind of the direction that I hope we're going.
Great. And two last questions. Erika, again, I think you addressed this in the in the chat, but what strategies has Penn State used to fund its microbiome research at the intersection of One Health?
Nicole could help here too, but we have some seed funds. So Penn State is really supportive of interdisciplinary research when it comes for a seed funding type situation. So the Microbiome Center has some seed funds. The Huck Institutes for the Life Sciences have some seed funds. We have seed funds in the specific colleges, but that's just to get stuff started. Unfortunately, we still reliant on the domain area, if you will, funding agencies. And the way we're managing to incorporate One Health into this is to have the main goal of the research cater to the funding agency that we are applying for, but also put secondary objectives that start getting us closer to the One Health. This is one thing that we are advocating a lot because there isn't really a funding mechanism to look at things from a multidisciplinary, like holistic perspective.
Great. And then our last question, what makes Penn State such a great place to pursue antimicrobial resistance research? And that's for anybody.
I just think we're cool and we help each other. And
I would say that as a new professor, having all the microbiome center is a big interdisciplinary. That's kind of a big thing. And I really, it's not just, in my case, it's not just plant microbes. I can see the whole picture in the institute, in the center, sorry. So that's kind of why, in my case.
Too is being a land-grant institution, right? We have a pulse on what's going on in real world and are able to have those conversations with farmers and try to address like ongoing practical issues that they have. So we spend the gamut on evolutionary studies where you're playing with bacteria in a petri dish to see what they do, all the way to a farmer has an outbreak. Like we're working with a farmer right now on Salmonella Dublin and trying to help them curb the outbreak on their farm and testing approaches and stuff like that.
Yeah, and then I would just add that we have a lot of research facilities and support. We have basically every type of sequencing you can think of, metabolomics core, proteomics core, and so on, fermentation facility. So not only do we have that expertise and community collaboration spirit, but we also have the actual tools to get the research done.
Wonderful. All right. Well, thank you guys for your time. And again, you can find this recording on Exchange in the next week. We appreciate all our panelists for sharing their recent work and, you know, all the impactful things that you are doing in the world of AMR. Thank you very much. And that concludes our member spotlight. Thank you.
Thanks everyone for taking the time.